Hello, everyone, and welcome to today's webinar titled GenScript's Solutions for Advancing the Development of Neoantigen Vaccines. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a few minutes to answer the survey questions. It will be available during both the live session and on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. All right, now let's get started. I'd like to present today's speaker, Melody Chang, Segment Marketing Manager at GenScript. For a complete biography on our speaker, please find it on the left of your screen. Melody, you may now begin your presentation.
Thank you. Hi, everyone. I'm Melody Chang, and thank you for the introduction of me and the GenScript company. And today my topic is GenScript's solution for advancing the development of neoantigen vaccines. I'm going to talk about three parts. First is neoantigen vaccine introduction. And the second part is the importance of neoantigen peptide. And lastly, we'll talk about the key considerations for mRNA vaccine outsourcing. Okay, let's first talk about the neoantigen vaccine. Let's quickly see this concept first. Before we talk about the neoantigen vaccine, let's take a look at the tumor antigen. There are two types of tumor antigens. One is called tumor-associated antigen. The short form is TAA. Another is tumor-specific antigen. The short form is TSA. What's the differences between them? For TAA, it's a high expressed in tumor cells, but also has some low expression on healthy cells. Previously, TAA is the antigen for researchers to use to develop vaccines, but it didn't very successful since 1999. The reason is because patients have central immune tolerance for TAA, so it's not an ideal antigen. Because of the AI technology and also because of the NGS sequencing technology, we got a chance to find TSA. It can only be found on tumor cells, but not on healthy cells, which makes it a very good potential for the treatment of tumors. Another advance is that it has a highly tumor-specific and has strong immunogenicity, better MHC affinity, and not affected by central immune tolerance. So this TSA gave us hope to develop a new kind of vaccines. And neoantigens is actually a kind of TSA. It's a type of it. It's classified by expression frequency. If it's very highly expressed, it's called shared neoantigen. But most of the cases, the vast majority of mutations are random and unique to individualized patients. So when we talk about neoantigen vaccines, mostly it means it's personalized. And of course, since it's personalized therapeutic cancer vaccine, we're trying to find the specific antigen instead of the associated antigen. And for the shared therapeutic cancer vaccine, mostly we're talking about the associated antigen. Only a few researchers are working on TSA because, it's very unique to find out. And shared therapeutic cancer vaccines are off the shelf and personalized. It has to be customized. And new antigen vaccines are used for cancer treatment. So first, let's take a whole picture and see the cancer treatment. It has a traditional way in which we use the chemotherapy, radiation, or surgery. But recent years, we're trying to use our own immune system to fight with cancer. It's called cancer immunotherapy or immuno-oncology. There are 4 strategies, and therapeutic cancer vaccine is one of it. And another ways are antibody drugs to block immune checkpoints, like anti-PD-1, anti-CTLA4, and adoptive cell transfer, like CAR-T therapy or TCR-T therapy. And the rest of it is non-specific immune stimulation. And new antigen vaccines are usually used combined with other therapies. So it's called combination therapies. Sometimes we will see some cases later, it will combine used with the immune checkpoints inhibitors. Okay, now let's take a look at the whole process for new antigen synthesis. Generally speaking, there are several steps listed here. First, you collect the specimens. It's called tumor biopsy. And then we do 3 levels of sequencing, like a whole exome sequencing and also RNA sequencing. Then we're trying to predict the new antigen, and after we sequenced the peptides. Finally, after the sequencing part, we got to do some evaluations of immunogenicity for the candidate new antigens. After we verified all of these things, then we decided to manufacture the vaccine. There are usually 3 types of vaccines for new antigens. And one is the synthetic long peptide vaccine. Another, we use RNA vaccine. And the last one is dendritic cell vaccines. Lastly, we'll do the treatment and tracking the results. So during this whole process, there are 2 key problems need to be solved. One is to identify the new antigens efficiently, accurately, and quickly, because the new antigen is the target, right? And it's very important to make sure it's the right one we want. And the second problem we got to solve is to find adaptive production process for different sequences in a short time and with a high successful rate, right? Because it's personalized, we cannot do it like the off-the-shelf therapeutic cancer vaccines to prepare everything ready. We have to get the sequence, then develop the vaccine quickly in a short time and with a high successful rate. So that's the 2 important problems we got to solve. Okay, another part we got to consider about the new antigen regimens is like this, and time to manufacture, and the vaccine dosing regimen, and combination therapies. Like I mentioned, the main kind of vaccine types are an RNA peptides and dendritic cells, but also we have viral vectors. So here, it listed the time to manufacture from short one and the long one. When the researchers, our customers, they choose the vaccine platform, time to manufacture is an important thing to think about. also the vaccine dosing regimen, like how many doses and when to have the dose, stuff like that. And once the disease reoccurred, we have to repeat the sequencing of the DNA tumor DNA, and also assess the vaccine-induced T-cell response in blood and tumor. And combination therapy, I mentioned previously, is for a good environment for when the patients got the vaccines. Okay, I have given us some introductions about new antigen vaccines. And next part, we'll talk about the importance of new antigen peptides. So there are two important reasons to use new antigen peptides. So first, let's take a look at the screening and validation process for tumor new antigens. So first, we will use the AI to predict the new antigens after several rounds of sequencing. After sequencing, the important part is to verify the immunogenicity. And lastly, we got the validated immunogenetic new epitopes. So from this picture, we can see from the beginning, there are about 28,000 of exome variations to finally only 3 validated new antigens. So how we validate it? Usually, we will choose ELISpot assay. We will use either peptide library or peptide pools incubated with the PBMC. And then we will stimulate it and try to detect the cytokines secreted from T cells. So that's why it's important to choose new antigen peptide library or pools. It's because the validation process is very important. If you just predict it, that's not enough. We got to do some experiments to validate it. And the GenScript new antigen peptide solutions, like is listed here, we can you can find the free design tools online, including these 3 listed here, and we have 7 in total, and these 3 are usually common for vaccines when making the peptide library design. And after you designed it, you have another tool to choose and to evaluate the difficulties for synthesis. After the evaluation, we will choose different methods to synthesize the peptides. So you can have a high successful rate to get your peptides. And lastly, we have several synthesis platforms. For example, this one is a high throughput, fully automated platform. It can provide up to 600 peptides at the same time. We also care about the quality control because, you know, the peptide libraries, there are usually several peptides together. And if you lose one, it will make your experiment failed, right? So we are caring about the quality. We're trying to ensure that 100% of the peptides in the pooling services are synthesized and mixed correctly. And we have providing the environment control like this C-grade clean room. And also we're controlling the cross-contamination to avoid false positive results due to the cross-contamination. Okay, we had talked about one of the reasons why new antigen peptides are important. Another reason for why it's important is because the new antigen peptides plays a key role for the development of new antigen vaccines. So, like I mentioned previously, the typical vaccine types are listed here. DC-based vaccine, synthetic long peptide vaccines, and RNA vaccines. And for the peptide vaccines, of course, peptides are used as the active ingredients, directly used as drug substances or drug products. And for mRNA or DNA vaccines, peptides are served as positive controls for vaccine screening and immunogenicity verification. The common forms for the neoantigens are the neoantigen peptide libraries or custom mixed peptide pools. And for DC vaccines, peptides are used as a crucial raw antigen material. It will be incubated with the DC cells and then preload a new antigen on DC cells. So it's very important, but actually it's not easy to be produced. And the reason is listed here. We'll consider the length, the charge, hydrophobicity, yield, and purity. So for new antigen peptides, if it's over 20 amino acid, it's difficult to synthesize. And the charge and hyphobicity, it will affect the purification. And the yield is the amount of how much we can produce. It's also another thing. It's not easy to do. And the GenScript solutions listed here, we have a streamlined workflow. First, we will have a new the trademark is called new pre-forecasting software to evaluate the difficulties of how to evaluate how difficult the peptides can be produced. Then we will synthesize and purify them under the PepPower platform. And also we have some industry-leading instruments to ensure we can provide more than 98% synthesis success rate, and we can provide high quality products as fast as 1 week. So we have talked about the new antigen vaccines, starting from the discovery period, like antigen identification. Then you have to produce the vaccines, then commercialization. And we can provide you different grades of peptides. For example, here, RUO and cGMP grade, and I listed the difference between them. For example, the facility is different. The cGMP one, we have the grade C suite and grade A isolator. Okay, next, let's see this case study by using the neoantigen peptides. It's a clinical application of preventing recurrence after surgery of solid tumor. 6 to 20 neoantigen long peptides were designed for each patient. Then neoantigen peptides are divided into 1 to 4 peptide pools. Each peptide pool contains 1 to 5 long peptides. And this customer purchased the GMP-like grade peptide, and the purity is more than 98%. And here's the endotoxin level. Finally, 10 patients received the vaccination after surgery. Among 7 patients who received the complete vaccination, 5 had the new antigen T-cell response. And no obvious adverse reactions were seen after the vaccination. The final result is the median recurrence-free survival was significantly higher than that of the other patients. Another case is a recently published paper, in May 2023. This customer and topic is the paper topic is the personalized RNA neoantigen vaccines stimulate T cells in pancreatic cancer. So the target is neoantigen, and the format is RNA vaccines for the treatment of pancreatic cancer. During this process, this customer is BioNTech and Kettering Cancer Center. They used the new peptides from GenScript to do the in vitro stimulation. And take a look at some details first. After the surgery, the customer used this anti-PD-1, PD-L1 immunotherapy to give the tumor a good environment to have to use the vaccine later. So here's the priming dose 1 to 8. Then how can we get this dose? It's because after this surgery, customers quickly sequence the tumor and the normal DNA and predict and select new antigens, then quickly manufactured the mRNA format neoantigen vaccines. And the delivery particle is lipoplex. So after this priming dose, then there's a 4-drug chemotherapy. Then later, another booster dose. So this is the whole process. Okay, I have explained the importance of new antigen peptides. Next part, let's talk about the key considerations for mRNA vaccine outsourcing. When we talk about mRNA, before this mRNA vaccine become very famous, it's actually the researchers have been developing it for many years for cancer treatment. But since COVID-19 happened, and researchers quickly adapted the mRNA platform to develop mRNA COVID-19 vaccine, and it validated a good success. Then this gave us more confidence on developing mRNA vaccine for the tumor treatment. So that's the importance for mRNA vaccine. And when we want to produce it, there are several key considerations. Here is listed. And when mRNA, we want to have a good mRNA. Actually, if you cannot have it in a good quality, it will affect the whole developing process. And the goal for develop a good mRNA is actually to have one very stable and cannot cause immunogenicity by itself, and finally can have a high protein expression rate. And here listed the blue letters listed several parameters that we can adjust to have a good quality mRNA. So the key considerations are listed here. First is the plasmid DNA or mRNA integrity. The logic is quite simple. If you want to have a good mRNA, you should first have a good plasmid DNA, right? It's the starting material, and it will affect the yield and the expression efficiency later. And in GenScript, we are the #1 gene synthesis companies, and we have a platform called GenScript's to guarantee the good codon optimization, and also ensure the plasmid DNA integrity. Another key consideration is poly-A tail quality. The length affects the stability, and we can build the poly-A into plasmid first. And the third consideration is mRNA capping. It also affects the expression efficiency. And we are offering cap 0, cap 1, and cap 2, and provide 97% capping efficiency. And the fourth consideration is the uridine modification. We are providing different kinds of options for NTP. And next one is the purity. We have optimized the IVT process to ensure a high quality product. And also we have desalt or oligo DT purification. This is the way to purify the dsRNA. The next one is the lipid nanoparticle packing. This lipid packing is for the prevent the degradation and also enable the cellular update and release. Finally, the manufacturing workflow is also very important. It will affect the turnaround time and quality. And we are providing a one-stop service with integrated production workflow. And for mRNA service, the manufacturer's site is in Seattle. Okay, I just gave you a general idea. Next, let's take a look at some details. First is about the plasmid DNA. Linearization is necessary for the in vitro transcription. So first, we use a T7 RNA polymerase. The transcription reaction continues to the end of the DNA template. If it's a circular DNA, it's like no beginning and no end. So the product always contains many unnecessary impurities. Then we use an enzyme to cut the plasmid into a linearized way. So it ensures the transcribed RNA of defined length and sequence. And why do you have to outsource the plasmid DNA? From GenScript, here's the comparison between the self-made linearization and linearized by GenScript. On the left side, it's a self-made linearization. We can see the obvious miscellaneous bands. And on the right side, it's the same circular plasmid linearized by GenScript. Next is the poly-A tail solutions. Poly-A tail can promote translation and prevent mRNA from degradation. Usually, when the length of poly-A tail is more than 80, it can play a relevant role. So in GenScript, our tailing method is to clone poly-A into the plasmid template. And the length of the poly-A tail on the mRNA, the quality depends on the plasmid template. Next, let's see the capping. So there are three ways for capping. There are There are two ways for capping. One is a two-step using the enzyme. Another is one step, is the co-transcriptional capping. And GenScript is using the one-step capping. Here it shows the high efficiency. It's more than 97%. And we have three options for you: cap 0, cap 1, and cap 2. Next, let's talk about the mRNA modification. And why do we have to modify the nucleotides? Because it can reduce the immunogenicity of foreign mRNA and also can enhance the mRNA stability and improve the later translation efficiency of mRNA. And actually, there's the controversy whether we should modify it. But Moderna and BioNTech developed modified mRNA products that have more than 90% effective at preventing infections. But the leading unmodified mRNA vaccine was 47% effective. So some other pharmaceutical companies who used to use unmodified has changed to use modified mRNA. And GenScript can provide a variety of chemically modified MTP options. Next, let's talk about the purification. Here listed the high-quality IVT mRNA standard, the plasmid and dsRNA protein and endotoxin level. And if the plasmid impurity plasmid was involved, it's gonna have a potential to cause cancer. So we got to purify it. And in GenScript, we have two ways. First is we offer crude purity. We use oligo-DT to remove all those residues I mentioned. And another is purification is we use several methods. And for example, anion exchange to remove dsRNA. And here we can see the picture of the results. So GenScript can provide you a one-stop solution from gene synthesis to mRNA synthesis, and finally the lipid nanoparticle delivery package. It's the last consideration we mentioned. It's about the workflow. It can enhance the whole efficiency, and also it's like a one project, so you can easily communicate with your project managers timely, and they also understand your needs pretty well. And also we reduce the risk of handover to several suppliers, and maybe it's also very time consuming. And when there are some problems, you don't know which step is the reason. So let's take a look at some details about this mRNA service package. For you, you just need to send us your ORF clone sequence. Next part are all for us. We will start with gene synthesis to linearize the plasmid. Next is IVT mRNA production. Here are some options for some key parameters. Next is the LNP formulation for RNA projects. Different mRNA applications need different delivery vehicles, and GenScript is providing you tailored LNP formulations for your vaccine delivery strategies. Also here listed some options for different parameters. Okay, so I've also introduced you guys about the neoantigen vaccines and the importance of new peptides, and also the key considerations when outsourcing mRNA. Finally, here I just show you a GenScript vaccine development workflow and solutions. We mentioned only new antigen peptides and mRNA service previously. Here you can see more details about other services options. So from the antigen discovery, step 1, it's for antigen identification and selection. We can support you for your virus vaccine or tumor vaccine study. And next, when you identify the antigen and want to develop your vaccine, we are providing you the solutions for the novel vaccine development, including DNA vaccine, mRNA vaccine, recombinant protein vaccine, a viral vector vaccine, and peptide vaccine. Next, for the vaccine effect evaluation steps, for example, preclinical study, usually it's for the cellular immunity evaluation and humoral immunity evaluation. We also have several options. For example, the peptide pools I mentioned previously, and also we can support you about the antibody development and labeling services for example, your ELISA assay. And also for some vaccines, you should test the antigen. We have a total solution from precast gels to a protein transfer system, which is called eBlot, and also an automatic Western blotting device called EasyVest. So lastly, the clinical development stage, we are also offering several kinds of products to support your analysis, and also services to support your manufacturing. For example, GMP plasmid DNA and GMP biologics. Okay, that's all for my presentation today.
Wonderful. Well, thank you so much, Melody, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. All right, so let's get started. Our first question is, do you provide any additional services related to IVT mRNA? For example, can you guarantee the poly-A tail length?
Yes, like I mentioned in my presentation, poly-A tail is a very important considerations, and we do have a poly-A guarantee service, and we can provide you high quality poly-A tail that are sequence verified and also customizable for your individual needs. In addition to that, we recently also launched circular RNA. It's a novel class of functional RNA. It's more stable and has a longer expression duration than the linear RNA I mentioned in my presentation. And we launched this service last month. And also self-amplifying mRNA, we are also, it's a new trend. So we are actually right now recruiting the self-amplifying RNA beta testers. So if you are interested in it, please contact us.
Great, thank you. The next question we have here is, when do I need cGMP peptides and what is the turnaround time?
Okay, so for cGMP peptides, In my presentation, I showed you guys the comparison between the RUO grade and GMP grade. And when can you choose the higher level GMP grade? My suggestion is you can start choosing GMP grade from the preclinical stage, and then use it for your clinical phase 1, 2, 3, and also the commercialization. And GenScript GMP facility is located in Zhenjiang, China. And right now we have 2 manufacturing plants. One, it's already launched. It's a grade C manufacturing site. And in Q1 next year, 2024, we'll have the phase 2 project, which will launch the grade A isolator within the grade C manufacturing background. It's used for the final step, finish step, like sterile filtration and aseptic filling for the product. And regarding to the turnaround time for GMP neoantigen peptide, generally speaking, it's about 30 to 40 days. And right now, there's no waiting time required because we have a big capacity. So just if you are interested in it, you can also contact us. And after we confirmed your order, the productions will take place once we got your order. Yeah.
Great. Thank you, Melody. Looks like we have time for one final question. Can you support IND work?
Yes, we support IND and also the CMC procedures. We can support your regulatory authority interactions, for example, pre-IND meeting or scientific meeting. And also we can support to response to the regulatory questions. Yeah.
Great. Well, thank you, Melody. This does conclude our session for today. Do you have any final comments before our audience?
I really appreciate your time to listen to my presentations. And if you have more questions from me, you can either contact me or contact our companies. Thank you.
Well, thank you again, Melody, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.